What steps are required to make a very resource-heavy industry greener? Does it make sense from a strictly economic point of view to reclaim and recycle, for instance, relatively cheap resources such as water and ethanol in pharma production? And if yes, how can it be accomplished? And what about other, less accessible process components such as buffer salts? A consortium of 3 Danish companies with competences in pharma development, water technology and production design took up the challenge. Traditionally, the pharmaceutical industry has not been very green. The only thing that matters is getting to market as fast as possible. Green solutions are not prioritized in the development phase of a new medicinal product. When you get closer to the market, you do not change your already established process, even though it might be better for the environment. Implementing green solutions and circular by design principles in the pharmaceutical industry has to be considered from an early development phase, especially in the concept planning of the processes of the production. We believe that our project may provide not only an example of making the circular concept an integrated part of the design process, but also can deliver concrete technology solutions that independently can be scaled broadly in the pharma and related industries. Here at Mipsilus, we are developing a medicinal product, Filimine, for a serious, rare metabolic disease called phenylketonuria, or PKU. The product, Filimine, is a new type of pharmaceutical based on a suspension of polymer-based nanoparticles that bind dietary phenylalanine, which is not tolerated by the PKU patient. Our product production consists of several process steps, which include 1. A polymer synthesis, where we make our polymer as a water in oil emulsion. 2. A wash of the polymer using ethanol and water. 3. Downsizing of the polymer using a bead mill to create our nano-sized polymer particles. 4. A subsequent wash using water. And finally 5. A chromatography process to purify the best binding polymer particles. The chromatography process uses a lot of phosphate buffer, which includes water. For most of our processes, we saw massive potential in both recovering and reusing water and solvents, reducing or completely eliminating resources used, and in-process integration of circular reuse of solvents. It was crucial for us to be forced to think about how we could spend our resources better. What solvents could be reduced and by how much? Was it possible to eliminate a process step altogether? And what consequences, if any, did it have to our product? Often it led to an optimization of the whole process, making the process greener and more time and cost efficient.
The synthesis of the raw polymeric product requires significant amounts of synthesis oil for the water in oil emulsion. After synthesis, the synthesis oil is reclaimed via filtration in a candle filter process that we have developed in the project. The candle filter unit is equipped with a hydrophobic membrane with a small cutoff that allows the separation of the raw polymeric product and passing of the synthesis oil. The process is operated in semi-batch mode, where the suspension from the synthesis is placed in the filtration unit and a filter cake composed of the raw polymeric product is built up on the membrane. At the same time, the oil is recirculated until the synthesis oil is clear. The separated oil is collected and stored before reuse in the next synthesis batch. And through this, around 80% of the synthesis oil can be reclaimed for reuse.
After oil separation, our raw polymeric product goes through several downstream processes in terms of washing, bead milling and chromatography. The washing steps require large amounts of pure ethanol and water of pharmaceutical quality. At each stage, we collect the ethanol and water fractions separately for subsequent recovery. Our chromatography step requires initial coupling of the chromatography resin with an expensive ligand added in 5 times excess to time efficiently couple the resin. The excess ligand is washed with water of pharmaceutical quality, and the ligand containing water is collected for ligand recovery. Additionally, the operation of our chromatography step requires large amount of phosphate buffer, a globally limited resource for the elution of Filimine and residual product. After separating Filimine and the residual product, the spent phosphate buffer is collected for recovery.
We have developed a customized decentralized reverse osmosis unit for the recovery of both ligand and phosphorus buffer. The recovery of ligand requires special operating conditions to maintain alkaline conditions, as acidification would cause precipitation of the ligand that would make it unfit for reuse. The concentrated ligand and phosphorus buffer solutions are stable and can be stored before reuse in the next processing sequence. The water fractions from the recovery of ligand and phosphorus buffer are collected for subsequent recovery in the water reuse unit.
The MIPSALUS project is cooperating with the EUDP project called EcoDist. The aim of the cooperation is to develop and test a flexible distillation technology for use in small and medium-sized pharma production facilities in order to make on-site recycling of solvents a realistic possibility. We have built and operated a prototype. This has informed the design and construction of the demonstration equipment. Through test runs, it has been possible to reduce the number of variables for the demonstration equipment. Safety has been a dominating issue in the design of the demonstration plant, since explosive vapors are processed indoor. In order to reduce the safety issues of the demonstration plant, it will not be equipped with vapor recompression, which was one of the two features which would provide energy efficiency. The distillation equipment should be energy efficient. It will be investigated further as part of the EcoDist project. It should be operating on electricity and not need external cooling systems. The equipment is operating on electricity, but since the vapor recompression has not been installed, some external cooling will be necessary. It should be able to run on start-stop operation, so not needing continuous usage, and the prototype needs less than an hour to warm up. The demonstration equipment is expected to have an even shorter startup time than the prototype. This makes start-stop operation easy. It should be possible to distill a broth containing solids. This has not been tested in this project since the liquids from MIPSALUS does not contain solids to an extent which will cause problems. But this type of horizontal distillation systems have been used for liquids containing solids in various industrial implementations. It should be modular built and horizontal and have a minimal physical footprint. A large number of variations can be made based on standard horizontal modules, and as can be seen, the equipment is quite small. The demonstration equipment will be even more compact. The fraction containing the distilled ethanol is called product, and the fraction without ethanol is called remanence. And we have been able to demonstrate nearly complete stripping of the ethanol with an ethanol content in the remanence of 0.1% and produce a product with an ethanol content of 51.5% from a feed with 17.5% ethanol. This was carried out with a capacity of 10.6 kilo feed per hour. If a higher ethanol concentration is desired in the product, the demonstration equipment can be configured to accommodate that, or the product can be rerun through the equipment.
Water of pharmaceutical quality is a key resource used in MIPSALUS' production of filamine. Water is used in all downstream processing steps in the purification of the raw polymeric product into the refined active pharmaceutical ingredient and for cleaning in place, so-called CIP, of the processing equipment. We have developed an intelligent water fraction collection procedure that basically divides the water into various categories of quality and treats it accordingly in our developed pre-treatment unit before reuse. The critical component in the reuse of the spent water is our central ultra-pure water treatment plant, which supplies the production with water of pharmaceutical quality. This has all the built-in barriers for removing residual impurities, not removed by the pre-treatment units. The water collected at the downstream processing steps will contain impurities that require removal to prepare it for reuse through the central ultra-pure water treatment plant. In particular, water collected from the washing and CIP steps will contain small particles as well as trace amounts of dissolved organic compounds that need to be removed before reuse. Our pre-treatment unit consists of an ultrafiltration unit and a UV-based advanced oxidation process to remove impurities critical for water reuse. Our concept for water recovery enables the reuse of 75% of the spent pharmaceutical water collected at the main downstream processing steps as well as CIP without compromising the quality requirements of the water produced for the production.
The current project has demonstrated that ambitions to make pharma production greener are realistic if the right initiatives are taken early in the process. By developing new technology and included in the process design phase, significant reduction of the value streams and reclaim of resources are possible. The technologies developed in this project are flexible and may find their way to other resource-demanding industries.